Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. If you like this video, subscribe my channel and click on bell icon to get notifications. In this video, I am going to explain the plasmolysis and deplasmolysis phenomenon. In a living cell, the behavior of a living cell regarding to water movement, it always depends on the concentration of surrounding solution. The solutions are basically three types based on its concentration, and these are hypertonic solution, isotonic solution and hypotonic solution. When we keep a living cell in these three types of solution, how the cell it behaves regarding to water movement, let us discuss now. In this, the first phenomenon is, it is called plasmolysis. So to observe this plasmolysis, by taking a living cell, and this living cell inside of it is filled with protoplasm. In this protoplasm, the cytoplasm consists of water and various dissolved inorganic and organic solute particles. In the structure of a living cell, by taking one living cell, in this living cell, the cytoplasm consists of water and dissolved organic and inorganic solute particles, and this cytoplasm, it gets certain water potential value. By keeping this living cell in hypertonic solution. So hypertonic solution having more number of solute particles, and due to having of more number of solute particles, its concentration is high, but its water potential value is low. So in this condition, by keeping a living cell in hypertonic solution, due to concentration gradient, the movement of water, it always takes place from higher water potential to lower water potential. That means from inside of the cell to outside of the cell, water movement takes place. It is called exosmosis. It is the process called exosmosis. So due to this exosmosis, water it is removed from the living cell. At this time, gradually the volume of protoplasm decreases. It is the phenomenon called shrinkage. That means, shrink of protoplasm takes place. The shrink of protoplasm is referred to as plasmolysis. So by keeping a living cell in hypertonic solution, the shrink of protoplasm takes place. It is called plasmolysis, and this plasmolysis, it is take place in three stages. In this, the first stage is limiting plasmolysis. So at beginning, when we keep the living cell in hypertonic solution, the shape of the cell, it changes slightly. So slightly changes take place, and it is said to be as limiting plasmolysis. Again, by keeping this living cell in this hypertonic solution for a long period, so gradually the water which is present in protoplasm, it will be came out. Then the water which is present in vacuole, it is came out from the vacuole into protoplasm. Then this water will be removed from protoplasm to outside of the cell. At this time, the shrinkage of protoplasm takes place, and protoplasm, it will gradually separate from the cell wall. That is, the stage is called incipient plasmolysis. It is called incipient plasmolysis. So in this incipient plasmolysis, protoplasm separated from cell wall, especially from corners. In third stage, it is called evident. It is called evident plasmolysis. So finally, when the water came out from the cell, the protoplasm it will be shrink, and this shrinked protoplasm it gets a spherical shape. Then it is settled at one corner of the cell. It is the stage called evident plasmolysis. So finally, the shrinked protoplasm present at corner, it is settled at one corner. Now it is the stage called evident plasmolysis. In this evident plasmolysis, in the structure of a living cell, the entire protoplasm in the form of a spherical shaped structure, it present at one corner, but in between this protoplasm and cell wall, empty space is present. This empty space, it is filled with hypertonic solution. In this way, gradually, the movement of water takes place from inside to outside of the cell. Gradually, protoplasm it will be shrink and settle at one corner. It is said to be as plasmolysis. So when you take some examples for this plasmolysis, the first example. So this plasmolysis generally we use in our daily life. The first example is salting of pickles and storage or preserving of storage of fishes and meat. Along with that, adding of sugar to jams and jellies. So in this way, we can add either salt or sugar to perform plasmolysis, especially in pickles. Due to adding of more salt, the concentration increases. If any fungal spores fall on this pickle at this time, due to exosmosis, the water which is present in that fungal spores, it will be removed to outside of the spore, and this spore it become inactive and it never germinate into fungus on pickle. That's why more salt is added to the pickle. In the same way, when the fishes and meat, by adding this salt, the plasmolysis can be takes place. Due to this plasmolysis, water will be removed from the cell. It can be stored for many days. Along with that, more sugar is added to the jams and jellies. It is completely based on this plasmolysis phenomenon. And one of the very important thing, the plasmolysis process can be demonstrated with the help of an experiment. In this experiment, one of the plant that is called Tradescantia. So Tradescantia is a plant. In this plant, the leaves are in different colors. In this Tradescantia, the cytoplasm is pigmented and it appeared with color under microscope. By removing the lower epidermis of the leaf, by keeping this epidermis layer with cells in a salt or concentrated solution, gradually the plasmolysis takes place. After few minutes, by keeping this epidermis layer under microscope, we can observe the shrinkage of protoplasm. With this experiment, we can observe or demonstrate the plasmolysis process. The next phenomenon is deplasmolysis. In this deplasmolysis, it is deplasmolysis. In this deplasmolysis, the plasmolyzed cell the plasmolyzed cell is taken. In this plasmolyzed cell, the protoplasm it shrink and present at one corner. In this plasmolyzed cell, the water potential value it is very low. Such type of plasmolyzed cell, when it is kept in hypotonic solution, in this hypotonic solution, due to having of less number of solute particles, the concentration is low, but the water potential value is high. In this condition, due to concentration gradient, the water which is present around the cell in hypertonic solution, it is transported from hypertonic solution to inside of the cell, from high water potential to low water potential based on concentration gradient. It is said to be as endosmosis. So in this way, the movement of water it takes place from outside to inside of the cell. It is called endosmosis. So when the water it enters into the living cell and when it reaches protoplasm, at this time the swelling of protoplasm it will be take place. It is said to be as deplasmolysis. During this deplasmolysis, the cell it gets their original shape. Again, due to concentration gradient, if more water it enters into the cell, at this time the protoplasm will increase, the volume will increases, and this increased protoplasm it creates pressure on its surrounding wall. At this time the cell it is said to be as turgid cell. It is called turgid cell. Due to entry of more water into the cell, the protoplasm it creates pressure on surrounding walls. That is cell is called turgid cell. This turgid cell, the protoplasm which creates pressure on wall, that pressure it is said to be as turgor pressure, and it is denoted with Tp. In this turgid cell, the wall which is present around the cell, this cell wall also with the help of some pressure, by creating some pressure on protoplasm, it protects the cell from cell bursting. The wall which creates pressure on protoplasm, that is considered as wall pressure, cell wall pressure, and it is denoted with Wp, that is wall pressure. So in this condition, when water enters into the cell, into protoplasm, at this time both this turgor pressure and wall pressure it become equal, but both are signed in opposite direction. It is the cell called turgid cell, and this process is called as deplasmolysis. In similar way, we have one more type of solution. It is called isotonic solution. So by keeping a living cell in isotonic solution, in this condition, the cell, especially cytoplasm which is present inside the cell, it having some water potential value, and this water potential value it is equal to water potential of isotonic solution. In this condition, the water movement it takes place from outside to inside and inside to outside of the cell freely. That is the cell considered as flaccid cell. It is called flaccid cell. So in this way, the behavior of plant cell can be observed regarding to water movement based on the concentration of surrounding solution with plasmolysis, deplasmolysis and flaccid cells.